talking about people who just do these things that might take 50% or 70% longer than the elite. Because the challenges that are faced with those individuals who are just participants and finishers versus the elite is quite different. They have so much longer period of time to address their nutritional needs. So we are dealing with the competitors, and we're not dealing with just the finishers. We're looking at some laboratory data, and of course anytime you have laboratory data, it doesn't necessarily represent too accurately, or perhaps it does, we'll see what happens out there in the field. We do have some field data, interestingly enough. In recent years, there's been a number of mechanisms available where we can catch real-time data in these very competitive individuals. We're dealing with humans, we're not dealing with particular strains of bred mice or rats, and humans are heterogeneous individuals. So one is going to respond differently to another, even though they may be equally well trained. And one may respond differently today than they might in a month's time, even though they have the equivalent level of training. So what we see in the following slides is going to be a general overview of a number of things, and it applies in general, but for any given individual on a particular day, it may not be that way for them, and that happens all of the time. So because we're dealing with these human endeavors, we have a variety of geographic environmental conditions that might be faced when people do these, which of course will have some impact on their ability to complete, and perhaps even what their nutritional needs are. Quick roadmap for what we're going to do, and we're going to see this throughout because as we go in through the different sections, we're going to highlight a variety of these. In general, we're going to look at what the performance levels are like in high energy demand endurance events. What's the energy cost? How do we determine that? What are the endogenous energy storage mechanisms? How much is available? Lipid oxidation, why don't we use it to a greater extent? What does training do to alter rates of lipid and glycogen utilization? How is nutrient intake a benefit? And how does the type and specific composition of nutrients have an impact on their availability or their bioavailability? So first up here, performance in high energy demand endurance events. What are these performances actually like? And I've going to focus mostly here on the World Championship Ironman event that just recently was held in Hawaii about 2 weeks ago. Here's the 2013 winner. They swim a 4-kilometer swim, 180-kilometer bike ride, a 42.2-kilometer run, and it takes them just over 8 hours. So that's 2013. 2013 female, fairly similar, 4-kilometer swim, just under an hour, 180-kilometer bike ride, under 5 hours, 42-kilometer run, 2 hours and 50 minutes, and they do this in a sequence, of course. Finish time 8:52. So that's 2013. 2012, I'll just point out here, AUS is an abbreviation for something. 2011, we have a Brit in there, okay. 2011, oh, we got another one of those up there. 2010, oh, another one. And male, oh, gee whiz, another one too. Let's go back to that one. I'm going to quit right now. Right, so what is the energy cost and how are these energy values determined? Well, if we just throw out some numbers from general knowledge about these types of activities and the rate at which they're done. Swim expends about 850 kilocalories. Right, that's a 4-kilometer swim. Takes them somewhere about 50 minutes plus or minus a little bit. Bike ride, 180 kilometers is 5,000 plus calories. And the run, 3,000 calories. That's a marathon at a pretty good speed. So the total expenditure for completing these Ironman events is about 9,000 calories, and if we take 8.5 hours as the time frame for that, it works out at about 1,060 kcals every hour. So it's per hour that's a critical equation here, or a critical relative measure to keep in mind, because there are limitations to how much we can take in, and those limitations are based on time. How quickly can the gut move stuff through? So keep in mind that a lot of the things we're going to look at are going to be per hour. Or at least as we look at stuff, keep that per hour relative measure in mind. Just by comparison, we've all heard of the Tour de France, and we know it goes for quite a long time, like about 21 days, and it has a whole slew of different things. Total time for an average Tour de France is about 92 hours. Their average speed is about 40 kilometers an hour. Their average power output is about 280 watts over that entire sequence of events, and of course they're going to be higher in some areas and lower in other areas. Their total energy cost is about 18 calories every minute, and that calculates out at about 1,080 kcals per hour. So whether we're looking at Tour de France or whether we're looking at World Championship Ironman performers, essentially you have about the same rate of caloric expenditure or energy output. How do we calculate these? So there are a variety of laboratory procedures that are associated with the measurements of metabolism in humans. There are a variety of procedures in other animals or insects as well. In humans, we tend to do this whole body organism through indirect calorimetry, where we collect all the expired air, we analyze it for oxygen and carbon dioxide, we make a bunch of calculations, and it gives us what the total energy cost is of moving at certain speeds. And if it's running, we can do it here on a treadmill. If it's cycling, there's a variety of different cycling devices that can be used. If it's in the pool, it's a little more cumbersome because obviously these are very large pieces of equipment that are being used. But here we have Dr. De Quisto, about 20 years ago when he was quite a bit younger as well, showing one of the mechanisms for collecting expired air in swimmers, and we hope they swim a straight line or he goes in the pool. And we then take that, oops, sorry, wrong way. We take that bag of air, we analyze it for volume and the concentration, fractions of oxygen and CO2, etc. We come out then with an aerobic metabolism profile. And this highlights for us what a typical aerobic metabolism profile would look like on a fairly well-trained, pretty good endurance athlete. Here we have workload. This is on a cycle. This is workload. This is power output in watts. This is oxygen consumption measured in milliliters per kilogram body mass per minute. And we continue through a spectrum of power outputs, each one being staged at a certain length of time. And the end result, when we get to a peak where O2 will typically start to plateau out, we can go more work. This is not the peak work that they can do. This is the peak level of oxygen consumption that we're going to get. If we're looking at peak power output, they might get up to 450 or 500 or 550 watts, but a lot of that's anaerobic power and not aerobic power. And you can't sustain that for an hour, 2 hours, let alone 8 hours, so that's irrelevant to this particular presentation. The other highlight on this is that we have an aerobic capacity here somewhere in that mid-60s. That's a reasonably high aerobic capacity, not the highest that's ever been measured. The highest are up here in the mid-70s, even up to 80. But it's much, much higher than your typical healthy but untrained individual. I'll highlight here the approximate percent of the aerobic capacity that's sustainable. And what we find when we look at real-time data is that these wattage outputs at 280, 290 power output levels are what are sustainable for those 5-hour bike rides. This is obviously coming from a bike, but that's what's sustainable for 5 hours for these highly trained individuals. You and I might be lucky to get to 290 for 30 seconds, maybe. but we're not these highly trained elite people either. We also get, from the lab, we get a substrate utilization profile, which has great importance here, because we are interested in getting a better understanding of the proportions of energy that are coming from lipid and the proportions of energy that are coming from carbohydrate. And there are very different storage capacities for each of those components. What we find, again, as we go across the workload spectrum here, and this is the same data set, we're just now breaking it out into proportion of energy that's derived from the oxidation of carbohydrate and the proportion of energy that's derived from lipid. And we pick that sustainable metabolic effort at about 280 to 290 watts of power. And we find that from a substrate utilization perspective, about 80% of the total energy production in the exercise here is coming from carbohydrate. In contrast, about 20% is coming from lipid. Okay? That's an important concept to remember as we go through. Let's look at some real-time data. This is a very busy slide, and it comes from GPS and power output data from the guy who happened to win the Hawaii Ironman in 2012. What I want you to focus on here is the purple line here, which represents power. Remember what we said from the laboratory data, sustainable power output about 280 watts of power. This guy's going for 4 hours and 30 minutes. And yes, there's some variations in topography that have to be addressed, which is why we have some higher power levels if they're going up a hill, and some lower power levels if they're going down a hill, especially if the downhill is somewhat long, and they get up to a velocity where they just can't continue to increase that velocity based on air resistance and the inability to keep pedaling. You know, once you get up to a pedaling rate of about 130, 125 revolutions per minute, the level of coordination starts to fall apart. And they just cannot continue to exert more power into the system, so there comes a terminal power level that's functionally appropriate for where they are. Point here with this very busy slide is if you take a look at this purple line, and if you just smooth that all the way out and took an average, you end up with 291 watts of power output. Average power output for the entire cycle. So what we came up with in laboratory projections is very, very, very close to what we come up with in real-time data looking at power output. Even more interesting is to take a direct measure of energy expenditure, wrong button again. Direct measure of energy expenditure using doubly labeled water. Doubly labeled water is a very expensive way of measuring total energy expenditure. It's about $1,000 a slug, and therefore this data is based on n equals 1. And it happened to be a good friend of mine who works at the University of Montana, who's an excellent triathlete, not quite world class, but he's certainly in the top 15 or 20 in the world in his age group, which was 35 to 39 at this point in time. So that's no slouch. And he did this when he did Ironman Kona in 2006. Not only did he do the doubly labeled water technique, he also took, believe it or not, a muscle biopsy of the vastus lateralis prior to and a muscle biopsy of the vastus lateralis after completing the event to look at glycogen depletion. So the guy is absolutely crazy. But what we find from the data here, doubly labeled water, is that they came out with a total energy expenditure for the duration of the event at about 9,000 calories. That's very, very close again to the laboratory projections that we came up with previously. So from real-time data of oops, how about we go this way? Real-time data of power output and real-time data of doubly labeled water confirm the data that we come out with from laboratory projections and laboratory measures. So what's the endogenous energy storage mechanisms? How much energy is available from each? If we look at glycogen very quickly, there are two tissue masses that have glycogen storage, the liver and skeletal muscle. The liver will hold about 80 to 100 grams. That's about maximum for it. Skeletal muscle will hold about 400 grams, maybe a little bit higher in extremely well-trained muscle, not nearly that much in sedentary muscle. And the total amount of energy value of stored glycogen with the 500 gram mass is about 2,000 calories. That's not very much when you consider that these individuals are expending, 9,000, 9,500 calories. In contrast, lipids have tremendous storage capacity, and having a 2,500 to 3,000 gram mass of adipocytes for energy storage, that's an extremely lean person. Okay, so but again, we're talking about these highly trained elite athletes. This is about a 4%, 4.5% body fat level. Okay, they weigh about 75, 72, 77 kilos total mass, and we've got about a 2.5 or 3 kilo adipose level on them. That's not you, that's not me, that's them. So there's about 20,000 to 30,000 calories. So if only we could tap into that, that would be great. However, what we'll find out, what we found out from the laboratory data is that when you're working at that high of a level, only 20% of your energy is going to come from lipid, whereas 80% of it needs to come from carbohydrates so that you can maintain that level. You cannot work at those levels and utilize lipid as an energy source. If glycogen is so critical, does training increase storage, and why is volume still so limited? Well, does the volume of glycogen stored change with training? Yes, it does, and it changes considerably. Remember we said before that there's about a 400, give or take a little bit, mass of glycogen storage in skeletal muscle? Well, in active, healthy, but not particularly well-trained humans, that number is close to 300, 307, 325, 275, or so on. And so with the appropriate training and with the appropriate nutrient intake and nutrient balancing with the training programs in the days preceding these large events, that muscle glycogen can increase up to about 50% of non-trained values, and so we're looking at this about 400 grams. This is millimoles per kilogram dry weight of muscle, but when we looked at that 400, 450 gram value before, that took into account the enhanced glycogen storage that can occur with the training that these individuals do, in combination with the nutritional intake practices that they employ. So why don't we store more? Well, given that the hepatic tissue and the skeletal muscle tissue are the two storage depots, we have to look at what the functions of these are. Hey, liver cells do a whole pile of different things. You just can't shove a whole bunch of more glycogen in there and think it's not going to impact what the liver does. So glycogen storage is important for the liver because it controls plasma glucose, and that's very critical for CNS functioning. But that's one of hundreds and hundreds of different roles the liver plays. So you can't interfere with those other roles simply by shoving a whole bunch more volume in there, and the liver's not going to take it in anyway. Skeletal muscle, the primary function is to generate force. Those contractile elements, the actin, the myosin, the tropomyosin, etc., that's what muscle is supposed to do, generate force. And within the boundaries of the sarcolemma, the muscle cell membrane, you only have a certain amount of space. And that space is taken up by contractile elements and mitochondria and enzymatic pathways for glycolysis and sarcoplasmic reticula and calcium storage and all of the different things that muscle does to generate force, or what it needs to generate force. So you just can't go shoving a whole bunch more glycogen in there, okay? Because it's going to interfere with the muscle's ability to do what it's designed to do, which is generate force. We don't have any other organ options, okay? There's nothing else that's going to be storing glycogen. Another reason we don't store more glycogen is because it's not a very efficient way to store energy. I know when you go to, what does 1 gram of glucose in terms of energy value hold? 4 calories. Okay, everybody knows that. What's 1 gram of lipid? 9 calories, 9 and a bit calories. Okay, everybody knows that. But when we store glucose as glycogen, for every gram of glucose that gets stored in there, there's multiple grams of water that are part of it. It's stored in a hydrous environment. Any calories in water? Not a single thing. Does it take up space? Yeah. Does it have mass? Yeah. So if you have 3 grams of water and 1 gram of glucose, how many calories do you get out of that 4 gram mass? 4. So you get 1 calorie per gram. So if we tripled, for example, if we tripled the amount of glycogen storage from 2,000 calories to 6,000 calories, we'd have a 4 kilogram mass increase. And for people who are performing these types of tasks, mass equals energy cost. And you cannot increase mass without having a cost attached to it. Besides, skeletal muscle is not going to do it, but it just highlights for us the inefficiency of storing energy substrates as glycogen. Does training enhance rates of lipid oxidation? And why isn't lipid oxidation utilized more extensively? Well, here we have the fat cell and the triglyceride in the fat cell, and this is all the storage mechanisms associated with it. And in order to get the fatty acids and glycerol released here from the triglyceride, they've got to be mobilized. There's a bulk transport in circulation. They have to get to the muscle cell and get diffused through the muscle cell. There's mitochondrial membrane transport controllers, and then we get to the oxidation level where we finally start to extract some of the energy that exists in these fatty acids. So it's a fairly complex sequence of events that begins at an anatomical location that is quite some distance from the skeletal muscle itself. So anatomically, it's not very appropriate, quite aside from the fact that every time it has to go through one of these processes, it's taking time, and the skeletal muscle doesn't have that time. So if we look at the mobilization aspect, here's our triglyceride. Here are the 3 critical signalers of hormone-sensitive lipase that break down the triglyceride into fatty acids and glycerol. The glycerol becomes a gluconeogenic precursor, gets picked up by the liver. The fatty acids in general circulation are utilized by skeletal muscle to produce, to go through oxidative processes and rephosphorylate the ADP back to ATP. Of these signalers, glucagon's a starvation-oriented hormone. You probably all know that. Doesn't have great importance in this particular presentation, although it could if they got into a total nutrient-depleted condition. ACTH, adrenocorticotropic hormone, is a stress response hormone. It's more of a chronic stressor. By far the most important signaler of this mobilization process is epinephrine, the catecholamine adrenaline hormone epinephrine, and it's directly related to the intensity of the work that's being done, and it's also related to the duration of the work being done. So the levels of intensity, that approximate 80%, is quite high. Epinephrine levels are quite high because it's a physiologic stressor. And the duration of the work, 8 hours, 8.5 hours, 9 hours, is quite long. Therefore, again, a physiologic stressor, high levels of epinephrine. Epinephrine is a very potent signaler of adipose breakdown and making these fatty acids available to skeletal muscle. The delivery process, okay, once it gets out of the adipocytes, it has to get to the muscle, forget about a lot of this other stuff here, and that delivery process is a function of cardiac output. what is the blood flow like that's going to be able to move these released fatty acids to the skeletal muscle? Now, most individuals, most healthy adult individuals, have a cardiac output maximum of about 20 liters per minute. Okay? Elite athletes have values somewhere in the vicinity of 30 or even higher, 35 liters per minute. So they have cardiac outputs. They can move 50% more or even 75% more blood each minute at maximum levels of cardiac functioning. That has everything to do, not with heart rate, but everything to do with stroke volume and the capacity of the myocardial contraction and the ventricular chambers to hold and pump blood every single beat. Their sustained, whoops, excuse me, wrong button again. Their sustained functional cardiac output is about 24, 25 liters per minute, which is, again, that's about 20%, 25% higher than the maximum capacity of a lot of healthy people. But they're sustainable for 8 hours, 9 hours, 10 hours. Then we get to the muscle tissue. And what's our sarcolemmal diffusion like? Well, to answer this question, there are a lot of factors associated with it, but one of the primary ones is what happens to the amount of surface area of the capillaries that surround the skeletal muscle that provide the opportunity for diffusion of these fatty acids. Typically, a skeletal muscle that is not well trained might have 1.5, 2 capillaries that surround it. Highly trained skeletal muscle has about 250% greater than that. So we're looking at number of capillaries around highly trained skeletal muscle, the GT trained, the GS sedentary here, of perhaps 5, 5.5, even 6 capillaries. More capillaries, more surface area, greater rates of diffusion, greater rates at which whatever is there in the circulation can get into skeletal muscle or get out of skeletal muscle if we're talking about metabolites. Mitochondrial transport. This is a mitochondrial membrane here, and here we have carnitine translocase and carnitine transferases, the outside portion of the mitochondrial membrane, and these are very important transporters for the fatty acids to get into the inner mitochondrial portion where they actually get oxidized and energy gets extracted from them. And so what we find with mitochondrial biogenesis and adaptations to training are tremendous increases in the mitochondria in general, and in concert with those increases are dramatic changes and increases in these carnitine transferase and carnitine translocase proteins that are very important for getting fatty acids into the oxidative process. Here we have the oxidative process itself and a representative enzyme of that, beta-hydroxyacyl-CoA dehydrogenase. We see dramatic changes in the concentrations of this with training. Okay. So bottom line with all of that is that lipid is an abundant source of energy, that there are tremendous adaptations that take place in the trained muscle. And those adaptations are both in the vasculature and in the enzymatic profiles of the muscle, and that lipid oxidation is substantially enhanced with these high levels of training, but even so the levels of performance still only draw about 20% of the total energy from the lipid compounds. Why don't we use it to a great extent? Well, there are any number of reasons, but one of them relates to the metabolic efficiency. If we look at the complete oxidation of a glucose molecule, and we look at what's called the P/O ratio, or the number of ATP that gets produced for every oxygen molecule that gets consumed, which is a chemical efficiency, that with glucose, we end up with 6 energy compounds for each oxygen molecule that gets consumed, whereas for fatty acids, it's about 5.6 energy molecules for each oxygen molecule that gets consumed. And you might go, well, 6 versus 5.6, that's not much. But it is 7%. And when you're working at a really high threshold level, 7% is a huge difference. When you're sitting here not doing anything, 7% isn't anything at all. Okay? It's nothing. But when you're working at that extremely high level, 7% is a major, major change. So, let's look at a nutrient algorithm here so we can move forward a little bit. Energy expenditure, 18 kcals per minute for 510 minutes. That's 8.5 hours. I just picked that number because it's somewhere in the middle of what the men and the women do. 9,200 kcals total energy expenditure. Relative contributions, 80% from carbohydrate. That means of that 9,200, 7,140 are going to come from carbohydrates. Total glycogen stored, liver plus skeletal muscle, 500 grams. So out of that 7,140, we have about 2,000 calories in the endogenous storage. We have a deficiency then of about 5,100 or about 1.3 kilos of glucose, 1,280 grams. So we have to somehow or another get that volume into the system and spread it over 460 minutes. Aha, you're saying, but we had 510 minutes up here, and now we've only got 460. That's because I don't know anybody who can eat food or that stuff when they're swimming. So we have to take into account that that first 50 minutes, they have no opportunity to take food in. So they don't get that opportunity until they get out of the water. That requires a consumption rate of about 2.8 grams every minute. Okay, doesn't sound like much, but we'll find out that that's actually a lot. So the conclusion from this algorithm is that carbohydrate intake is essential and that the amount that's internally available is severely limited. How does carbohydrate intake benefit? Well, let's look at one of the critical elements related to carbohydrate intake, and that has to do with the prevention of hypoglycemia. As you know, the brain requires carbohydrate glucose as not a primary energy source, it's a sole energy source. It doesn't have a choice. If you get into a hypoglycemic condition, your blood glucose drops down there below 55, 50, 45, 40, you're basically out, okay? And so if your brain is not working other than basic vegetative functions, nothing else is going to be working particularly well either. So the consumption of carbohydrate products over an extensive length of time versus a placebo is able to maintain plasma glucose at reasonably high levels. This is near 5 millimole. If you multiply that by about 18, then that's about 90 milligrams of glucose, 90 milligrams percent. So this is maintaining a euglycemic state, critical for CNS function. Here we're getting to a point now where we're down to, 2.5 millimole, 2 millimole. That's 2 millimoles, about 36, 38 milligrams percent of glucose. That is on your back, supine position, calling for help status. It also blunts the rate of endogenous glycogen diminishment in the skeletal muscle. So as long as we can continue to provide some glucose through circulation, then some of that is going to be used by the muscle, which means that the storage depot in the muscle is not going to be depleted at quite the same rate. Does the type and specific composition of carbohydrate intake affect substrate availability? Well, let's look at some of the different approaches that have been taken. Does it make any difference if we have solids versus liquids? And remember, we're talking about carbohydrates. We're not talking about combined food products that have a lot of protein and lipid and carbohydrate in it. And these are specifically the types of sport nutrient products that have been developed. Okay, so does solid versus liquid have, does it make a difference? No, not really, as long as we have good control over the volumes that we take in. As long as those volumes are equal, and as long as they're reasonable, we don't overload the GI tract, and yet we still provide enough, then whether you take it solid versus liquid, as in a bar, power bar, or Cliff Bar versus Gatorade type stuff, it doesn't make a lot of difference. If you take the same product as a banana and you puree it versus a solid, it's not going to make any difference. Your GI tract is pretty good at breaking down that banana, which is a reasonably high glycemic index food product. How about GI tract comfort? Does it make any difference if you have solids versus sport drink versus water? Yes, there's a level of perceived fullness if you have something that's solid versus something that's liquid, and you would expect that to be the case. Now, one of the elements that happens when people are out there for these 6, 7, 8, 9, 10-hour things is they do start to feel hungry. And part of that hunger sensation has to do with receptors that reside in the gut. And drinking liquids often is insufficiently strong enough as a stimulus to activate those types of sensory phenomena. And so eating something, solid products, be it bars, be it a banana, be it a Cliff Bar, whatever it might be, does alleviate that sense of hunger because you get some solid food product into the GI tract as opposed to just liquid products in the GI tract.
And that's still just just over half of the rate of energy consumption, though, isn't it? That you had for the recipe? Didn't you have 2.8 grams per minute?
The what?
Didn't you have 2.8 grams per minute for the Ironman recipe? And so there, I was just curious.
Here?
Yeah.
If you put what?
For the Ironman recipe, I thought you said you had to consume 2.8 grams per minute.
Well, I'm glad you remember that number. That doesn't mean that that works. That means that's what they need. Okay. What we'll find is that that doesn't work. Does the type of exogenous carbohydrate affect the bioavailability? Well, let's look and see what we've got here. We've got glucose, we've got polymerized glucose, we've got fructose and galactose, and we've got a variety of combinations of these. How do we know what the bioavailability is? Well, we use radioactively labeled carbons here with these glucose compounds. And so it can be measured in terms of when it gets oxidized, because it will come out as radioactively carbon dioxide. So it's measurable. So we put it in the system and we can measure its rate of oxidation through radioactive labeling. What's the, when we feed this rate, what's the rate of glucose oxidation here, or glucose polymers? It's about 0.9 or 1 gram per minute of oxidation. When we use fructose or galactose, it's about 0.6. When we combine glucose and fructose, we get up here to about 1.3 rates of oxidation. And if we dump even more into the system, 2.7 grams per minute, it makes no difference. Okay? We can keep shoving more and more food into the system, but if the system can't move it from the GI tract through splanchnic circulation to get to the liver, to then get to general circulation, there's no point in clogging up the GI tract and generating some osmotic effects there that could be distressing to the gut itself. So let's look at the limiting factors here. We know what the oxidation rates are like here, and we can see that if we have some combination of glucose and fructose, that appears to be the most advantageous. So where are the potential limiting factors here? Is it the GI tract itself? Is it gastric emptying? Is it liver processing? Is it the skeletal muscle uptake, or is it intestinal absorption? If we look at gastric emptying, and there's been study after study after study after study that looked at volumes and concentrations, and at the level of work rate of these individuals, 80% approximately of their aerobic capacity, and at the concentrations that are being given, there is no evidence to support the gastric emptying as being a limiting factor. In other words, at that about 1.7 to 2 grams per minute of intake, that it moves through the gut at the same rate as 1, as 0.5. Okay, so we've got a concentration here that works as far as gut emptying goes. What about liver clearance? They've done any number of studies that look at the movement from the hepatic portal system into the liver, and then its movement from the liver out into general circulation. No evidence to suggest the liver is somehow or another taking in this glucose and keeping it for something. It's not. It moves it right through and puts it out into general circulation. In large part because epinephrine is a very potent mobilizer of glycogen in the liver. And so you've got high levels of epinephrine already in the system. And that's a signaler to break down glycogen in the liver. So if the liver's receiving glucose from the hepatic portal system, there's nothing there that's going to signal the liver to start storing it. Not a single element. Skeletal muscle uptake. 95% of the glucose that passes through the liver entering circulation is oxidized by the active skeletal muscle through the radioactive isotoping, looking at the CO2 output. Trained skeletal muscle has enormous volumes of GLUT4 transporters, which are the translocase proteins in skeletal muscle that pick up the glucose from the outside surface of the sarcolemma, move it in, move it to glycolysis so it can start to go through aerobic metabolism. And so trained muscle has tremendous capacity through this whole GLUT4 transport mechanism to take what it's given, and it will use it. Okay, this is what we saw before, just to rehash, that it's a combination of glucose and fructose that appears to give us the highest levels. If we throw sucrose in there, it does the same type of thing. So why the difference? What difference does it make, and where's the basis for this? Well, as it turns out, the small intestine has different transporters for glucose and galactose than it does for fructose. So by taking a single compound, such as glucose, it's quite easy to saturate those transport mechanisms. Once they're saturated, you put more glucose into the system, it's not going to do anything. Similarly with fructose, if all you do is throw fructose into the system, you're going to saturate the fructose transporters. And so the best way to get maximum input through the hepatic portal system, through this translocation across the small intestinal membrane, is to do the right combination where you saturate both sets of transporters and get maximum possible delivery in through the hepatic portal system. The glucose transporter is a sodium dependent. It's a sodium dependent glucose transporter. There's a symport mechanism here. It involves some active transport of potassium and sodium in the middle of the interstitial cells here, and then it moves through into the capillary and the hepatic portal vein system that's then going to go up to the liver. So this highlights how glucose moves through the system, galactose moves through the same portal mechanism. Let's see. Okay. Wrong way. When we look at the maximum volumes of translocation then of this sodium-dependent glucose transporter and GLUT5, which is the fructose transporter, when we saturate the GI tract, this is about what we get through into the capillary system on the other side. We get about 60 grams of glucose per hour. And if we saturate it with fructose, and we saturate the GLUT5 transporters, we get about 30 grams per hour. So combining those two components, we get about 90 grams per hour of monosaccharide that's usable then in metabolism down the road. So when we look at these food products that exist, and we take a look at the composition, they don't tell us on their exactly what fractions they are, but they do indicate that there's combinations of glucose, polymers, and fructose. Okay. Now I can tell you that companies like Power Bar and Gatorade pay a lot of attention to this research, and they do a lot of the research themselves. Okay. So the products that they make are specifically designed based on what the research tells them in terms of GI tract functioning and bioavailability. So let's go back to our nutrient algorithm. This is what we started off with. We needed 9,200 kcals to finish at the rate that energy is being expended. Okay, we came down here and we knew we needed 2.8 grams of carbohydrate every minute. Based on our understanding of GI tract limitations and translocation, about the maximum we can get out is 1.3 grams per minute. That's the 90 grams, the 60 grams of glucose and the 30 grams approximately of fructose. And at 460 minutes, that leaves us a deficit of that amount. Okay, so we need that amount. We're only getting that amount, and that's why we're left with a deficit. So that's where the gut is the limiting factor in nutrient availability in these high performance tasks. So with that, I will try to answer any questions that you may have.
Questions?
Could you say something about ignoring protein as a caloric factor?
Consuming protein or?
Yeah, or endogenous if you're out there long enough.
There's a couple of lines of evidence related to protein consumption. One of those has looked quite extensively at whether protein in any way facilitates carbohydrate absorption or not, and the answer to that is no. What would the value of digesting relatively simple amino acids be? It could have a retarding effect on any internal catabolic processes of protein elements themselves. So that could be a potential energy source if we look at gluconeogenic pathways, alanine, pyruvate pathways there, where some of those amino acids could now contribute to the energy pool that's not being accounted for in glycogen and not being accounted for in exogenous carbohydrates.
So it's just really not important whether you consume amino acids or it could muscle break down some of the
Well, what we find,
amino acids can feed into the TCA cycles.
I mean, what we find within these products is some amino acid components that are thrown in there.
So I guess going back to the next slide, my question was, so what, or the other way, there you go. So how do these people ever finish this event? Mile 6 in the marathon, that's an Ironman, and they're on their back looking at the sky. I guess I've seen that happen.
Yeah, Jeff, I wish I had the answer to that question. I have a couple of hypotheses as to how they can do it. One is that they may, and while this data has come about from well-trained individuals. It has the GI tract translocation data hasn't necessarily come about from a lot of the best of the best. It's potentially possible that the gut is a trainable organ, that a highly trained gut with this system might be able to translocate 20% more, 25% more. We don't know the answer to that yet, but I think it's a potential. Another potential is that the rate of gluconeogenic processing by the liver, taking those glycerols that are coming out of the adipocytes, is higher than we think, and therefore that's a source of glucose. At 80% of aerobic capacity, there's going to be some lactate formation, but the rate of lactate disappearance is going to equal the rate of lactate appearance. So where is that lactate going? The Cori cycle. There's another gluconeogenic precursor that exists there that is not taken into account for here. So I think there are a number of possibilities. The gut is a trainable organ that we don't know about yet in this type of high energy demand situation. There's greater rates of gluconeogenesis occurring from glycerols, from amino acids potentially, and from the lactate that is going to be produced. And do they all occur? I don't know if the gut adaptation occurs, but the others do occur, but it's not easily measurable like these are easily measurable. So I think they would account for some of it. Do they account for all of it? I don't think so, so there has to be something else in the system. Do they slow down? You know, if you look at the splits, no, they really don't slow down very much. They maintain, when you have somebody running a 2:45 marathon, and their last mile is 5 minutes and 45 or 6 minutes, and their first mile was 5 minutes and 45 or 6 minutes, and that was after they just finished 5 hours or 4.5 hours of riding a bike 180 kilometers. They don't slow down. No. Yes?
If you look at one of these athletes and playing the numbers of that 9,200 kilocal expenditure, what happens when you look at what they actually ingest during the competition? Is there a gap of what they're ingesting and that figure?
Right.
It sounds like they're almost, you haven't shown any pathway, I think like Jeff was talking about, any pathway to get it in and actually get it to a usable form. There's this missile part. But are they ingesting?
So the question is, what are they actually ingesting when they're doing these events?
Yeah, and does it add up to a number like that?
Yeah. These athletes are very well tuned to the research on nutrition, primarily because they have individuals who are working with them who are tuned into it and they direct them what to do. They have very regimented nutritional intake procedures as they do these events. That is, every 15 or 20 minutes they do something, whether they feel like it or not. And one of the major principles of this is that you cannot let yourself get into a deficit, because with the limitations of the gut transport, if you don't start feeding until you start to feel bonky, you're way too late. I mean, you have to start feeding right from the outset. You get out of the water. In that first 15 minutes of the bicycle, you start feeding, and you do a regimented feeding routine every 15 or 20 minutes at these types of rates. And they have done analysis on the food that they take with them and what ends up back on the bike or in the bottles at the end, and they consume at right about the rates and that's as much as they can take in. If they take more, they get GI tract upset. They get the osmotic effect of that extra glucose, the extra liquids, whatever it might be. That's not a good thing, because then they've got to stop. Or they have to slow down and work out the GI tract.
So there's some mix of processes that turn those calories taken in into output.
Yes.
That aren't not completely understood.
Right.
Vince, are there athletes who give up their blood afterwards? So what's the average glucose level of an athlete glucose? So if they're 2400 calories that sort of implies that they would have pretty low glucose at the end of the
Oh.
race unless there's a compensating mechanism.
Right. So if I understand your question correctly, to share with everybody else, what's the level of maybe skeletal muscle glycogen like at the end of this? What's the level of blood glucose like at the end of it? The very few studies that have been done that look at muscle sampling, as one done by my friend, where the initial muscle glycogen level, millimole per kilogram wet weight of muscle was about 180, 190 millimoles per kilogram wet weight. The final value, a normal muscle storage, just so we have a comparative level here, a normal muscle storage is going to be about 100, 90, 100, somewhere around there, millimoles per kilogram wet muscle. The final value for this particular person was about 40. So they are well in the utilization part of that. What's blood glucose like? I don't have an answer for that, but I suspect it would be, I suspect it's maintained reasonably well because they're focusing.
They're not hypoglycemic by the time they're done.
Well, some of
On
them are.
average.
I mean, we've seen the hypoglycemic ones staggering all over the place, and they're the ones that make the highlight reels, but, yeah.
Just curious how it's measured, the 80%, 20% ratio between the carbohydrate versus lipid metabolism. How is that measured, and is it trainable, or is there any
Sure.
mechanism for people to raise the level of lipid?
Yeah. The question for people up the back that may not have heard that is, how do we come up with this 80%, 20% ratio? How is it measured, and is it trainable? When we measure aerobic metabolism by indirect calorimetry, we collect all that expired air and we measure it for fractions of oxygen and CO2. When we completely oxidize a glucose molecule, we produce 6 CO2 and we utilize 6 oxygen, so the ratio there is 1. When we oxidize a fat, depending on which fat it might be, but generally when we oxidize a fat, the ratio of CO2 produced, oxygen consumed is 0.7. So when we look at those profiles and look at what's called the respiratory exchange ratio of CO2 to O2, then we know what proportion of the total energy is coming from glucose metabolism and what proportion is coming from fatty acid metabolism. So that's how we measure it. The second part of your question is it trainable? Yes. Remember that, I'd have to go way back to the graphic, but if we were looking at an untrained individual, remember early on, about a third of the way through that, we had that crossover effect? Okay, that crossover effect would be much further out on a workload spectrum for an untrained person. Okay, so yes, actually it's the other way around. I'm sorry, it's the other, it'll move to the left. Yeah, so that the trained person does a much higher level of work before they get to a 50-50 ratio, and then of course it keeps going on from there. The untrained person, or the not well-trained muscle, which is not as well adapted for utilizing lipid, for example, it's going to tend to rely more on glucose at an earlier workload than a later workload.
For the top level athletes, they've already plateaued, they're not.
Yeah,
like
they're way out
change
there
the 80-20
on the right side of the spectrum.
What about genetic variation among individuals such that elite athletes perhaps come from a group of people who have genetic variants that either produce more receptors or receptors that bind more efficiently to,
Sure.
Things like that, or utilize
Sure.
them?
Well, the simple answer to that question is I think a lot of those Australians and Brits and others, are luckily the Kenyans can't swim or ride bikes very well. The more appropriate answer for it is, of course, there are genetics at play, and we look at that whole nature versus nurture component and functional ability of humans. Anybody who's the best in the world at what they do has a tremendous genetic advantage to start with. They happen to have had the opportunity in terms of training, in terms of economic circumstances, in terms of nutrition, in terms of medical support and help. They have had the opportunity to develop that genetic potential to the highest levels. There may be as many people out there, if not more, who have the same genetic potential, but just never developed it. And there are also so many out there who have the same genetic potential and don't have the opportunity to develop it.
But I mean, certainly there have been genome-wide association studies looking for alleles that would.
You know, the more and more we know about the way intramuscular signals occur and the different molecular components that are responsible for things like mitochondrial biogenesis, other transport mechanisms within muscle, regulatory components of metabolism, that there are certainly alleles and there are genetic components that are critical factors at play within that muscle structure itself, absolutely. Yeah.
Maybe one more question, and then people can individually come up and talk with Vince, so, yes.
So the Ironman in Hawaii, does the humidity affect respiratory exchange, and does it slow down metabolic rate?
Yeah, the question is, how does the humidity factor in Hawaii affect some of these variables? Respiratory exchange or the rate of energy expenditure? The short answer is humidity can have a tremendous effect in a number of different ways. One, the rate of sweating is typically relatively high, especially when the humidity is high, and sweating itself is not a mechanism for heat loss. It's the evaporation of the sweat that's critical. So the higher the humidity, the poorer the evaporative rate. So that's a factor. They lose water. They've got to perhaps consume a lot more water or fluid components than they would. Whenever you have tremendous thermoregulatory challenges, you have cutaneous vasodilation components that have to try to attend to that. And by attending to that, you only have a limited blood volume, a limited cardiac output. It's not an infinite number. That's going to channel some of that blood flow away from skeletal muscle to the periphery, to the cutaneous regions. And as soon as you take some of that blood flow, let's say it's 10% of typical muscle blood flow at high rates, it's now being rededicated to cutaneous vasculature. That's reduction in oxygen supply, it's a reduction in nutrient supply, it's a reduction in thermal transfer from the active muscle back out to get it out to the core. It's a reduction in CO2 removal from skeletal muscle. So the environmental conditions are going to have a tremendous impact on it. But you know, we've seen times in Hawaii that are world best time, 8 hours and 3 minutes in 2011. 8 hours and 3 minutes. That's a phenomenal effort. 8 hours and 10 minutes, 8 hours and 12 minutes, 8 hours and 15 minutes. Phenomenal efforts. So these individuals just consume liquids to the extent they can. They use ice sponges and anything they can do to keep themselves relatively cool. But yes, environmental conditions can have an impact on it.
So, 2 weeks from today, next seminar, and it will be, the title is Osteopathic Principles